Hello, everyone. I'm Xiaofan Qin, founder of GI Farming Inc. Thanks to the organizer for giving me the opportunity to present at this summit. Funding my research about 20 years ago led me to develop a great interest in gut bacteria and made me believe that impaired innervation of a digestive process due to a decrease in gut bacteria may have played a critical role in the pathogenesis of inflammatory bowel disease or IBD, including both ulcerative colitis and Crohn's disease and possibly multiple other health problems associated with dysbiosis of gut microbiota. Here, I would like to share my story and my thoughts. As we know, many diseases increase dramatically alongside industrialization and modernization. This slide shows changes in the incidence of IBD, including Crohn's disease and ulcerative colitis from 1930 to 2010 at different locations around the world. Although there are some irregularities, we can clearly see the general trend of a remarkable increase. This slide shows the increases of other diseases such as asthma, multiple sclerosis, and type 1 diabetes in the last century. Many studies in recent years have revealed that dysbiosis of gut microbiota has played a key role in the pathogenesis of IBD and many other diseases. This dysbiosis mainly manifests as decreases in both abundance and diversity of gut bacteria. Many of these diseases are also associated with increased intestinal permeability, the so-called leaky gut, and thus increased infiltration of toxins from the gut lumen into the body. Thus, the key question would be, what made the gut become leaky? Here, I would like to show you my explanation that impaired innervation of digestive processes in the lower gut due to a decrease in gut bacteria may be the primary cause. My interest in gut microbiome started 20 years ago when I found in 2001 that the digestive process such as trypsin and chymotrypsin can be inactivated by unconjugated or free bilirubin, but not conjugated bilirubin or bilirubin. Conjugated bilirubin, unconjugated bilirubin, and bilirubin are components of the bile. Trypsin and chymotrypsin are enzymes produced by the pancreas and released into the upper gut to help digest protein from foods. This slide shows the conjugated bilirubin and trypsin activity in the intestine of conventional and germ-free rats. We can see large amounts of conjugated bilirubin and trypsin can be found in the large intestine of germ-free but not conventional animals, suggesting bacteria are needed for the deconjugation of the predominantly conjugated bilirubin released from the liver through the bile into the gut, and also needed for innervation of a digestive process in the lower gut. The finding that digestive process can be innervated by free or deconjugated bilirubin, but not conjugated bilirubin, provides an explanation of the intrinsic link between deconjugation of the predominantly conjugated bilirubin and innervation of a digestive process mediated by gut bacteria. When we talk about bilirubin, most people probably think that bilirubin is bad, as high levels of bilirubin are associated with disorders such as brain damage and seizures in infants, or hepatitis or cirrhosis syndrome. However, in fact, bilirubin seems to be purposely produced. Bilirubin is the end product of the metabolism of heme, which gives the red color of the blood. The transformation of heme to bilirubin is a two-step process. First, heme is transformed to bilirubin by heme oxygenase. Then bilirubin is transformed to bilirubin by the action of bilirubin reductase. Bilirubin reductase even exists in some of the most ancient bacteria, like cyanobacteria, far predating the appearance of eukaryotic cells on Earth. This gene exists in almost all water-breathing animals. However, the transformation from bilirubin to bilirubin seems highly regulated. For instance, the bile of rats and mice is composed of mainly bilirubin, while the bile of a nutria, also a rodent, is composed of mainly bilirubin. The bile of trout is composed of mainly bilirubin, while the bile of tilapia is mainly bilirubin. People have long been puzzled by the physiological significance of this transformation of bilirubin to bilirubin. Bilirubin is innocuous, water-soluble, and readily excreted, while bilirubin is lipophilic, potentially toxic, and consumes more resources and energy for its transportation and excretion. The existence of bilirubin or bilirubin predominance in animals at different stages of evolution and closely related species suggests that the predominance of bilirubin or bilirubin is not determined by the currently well-known functions such as antioxidant activity or other intrinsic imperative needs, but rather adaptation to some environmental conditions. When we look into these species, the bilirubin predominant species are mainly herbivorous, while bilirubin predominant species are mainly carnivorous or omnivorous. Although the bile of cattle consider mainly bilirubin, the hepatic bilirubin reductase activity is only about 5% of that of rats. The main difference among carnivores, omnivores, and herbivores is the amount of protein in their diets. From the study shown here, we can see that the increased protein in diet increases pancreatic trypsin production. Therefore, carnivores and omnivores would have more digestive processes produced than herbivores, while inhibition of a digestive process by bilirubin would be the possible evolutionary driving force for bilirubin or bilirubin predominance in animals, as discussed in the paper I published in Gut in 2007. One question would be, why does the high amount of digestive process in the large intestine of germ-free animals or in the small intestine that have digestion of proteins not cause damage to the gut? The answer to the mystery lies in the special structure of the protective mucus layer. Mucin, the building block of mucus, contains a core peptide surrounded by many carbohydrate side chains that can only be rapidly broken down in the presence of both glycosides, mainly from gut bacteria, and proteins, mainly from the pancreas. Thus, a most pronounced degradation of the protective mucus and the damage of gut barrier may be at the intersection between the germ-free and conventional conditions, as discussed in the paper I wrote in 2008. Not surprisingly, the use of antibiotics would be one of these conditions, as shown in multiple studies that large amounts of digestive process appeared in the feces of people or animals treated with some antibiotics. When looking into the literature, I also found multiple studies showing that patients with IBD, including both autocolitis and Crohn's disease, also have much higher level of digestive process in feces. Studies also shown that patients with IBD have much thinner and defective mucus layer that allow gut bacteria to contact and infiltrate the tissue. Surprisingly, shortly after I found that the digestive process can be inhibited by achondroplasia bilirubin, I also found a study showing large amounts of digestive process in the large intestine of animals treated with saccharin in the diet. At the beginning, I did not know what saccharin was. After I looked in the dictionary, I learned that it is the artificial sweetener called tangjing or sugar elite in China. I grew up in a poor village in China and used the sugar elite when I was a child because it was cheap. However, my mom, who is illiterate, always warned me, be careful, the sugar elite is toxic. So we usually just took several granules each time. I wondered how commonly it was used in the US. Here, you can find it near almost every coffee machine and in every restaurant. People can easily purchase a box with 1,000 small bags in the supermarket. I was further surprised that multiple studies since the 1970s found that IBD patients have increased intake of refined sugar. The evidence was so strong that it attracted some scientists to look into the relationship between sugar consumption and IBD over time in different countries. However, they failed to find any meaningful correlation, and thus attention was gradually attenuated. These findings made me wonder, could IBD be associated with not refined sugar, but rather artificial sweeteners such as saccharin? This thought made me look more into saccharin. I learned that saccharin was discovered in 1878 by Konstantin Furberg, a foreign chemist in Professor Avery Ramsey's lab at Johns Hopkins University. At that time, they were trying to make new chemicals from coltan. One day, Konstantin Furberg noticed the more sweetness in the bread he was eating. He traced the sweetened back to a Turing chemical he worked with. Later, he named it saccharin, which is hundreds of times sweeter than sugar. Years later, Konstantin Furberg applied a patent for saccharin. After experimental production in New York, he built a factory in Germany in 1886 to produce saccharin. However, only the portion of saccharin was actually consumed in Germany, mainly due to the bad image of this coal-tar product in that country. Saccharin was regarded as being inferior and only consumed by people who could not afford the luxury of sugar. Saccharin was also prohibited or detested in many other countries, such as France and Italy. In contrast, saccharin was greatly appreciated in the United Kingdom. The founding of saccharin from coal tar was regarded as the most remarkable instance. People were assured by officials and doctors that saccharin was harmless and enjoyable. Shortly after its marketing, saccharin soon appeared in almost every family grocery in the UK. Although it was suspected that some forms of diarrhea described in ancient books could be sporadic cases of ulcerative colitis, it appears that cluster cases of UC only started to emerge after 1888. As stated by Dr. Sidney Phillips's discussion during the first symposium on IBD in the world in 1909, that presented a collection of more than 300 of UC patients from 9 hospitals in London. Quote, ulcerative colitis appeared to be much more common now in this country than formerly. There was no mention of it in any of the published reports of any of the London hospitals before 1888. The speaker himself had seen many cases at the St. Mary's Hospital and elsewhere since 1888, but not before then. In addition, Dr. Phillips even suspected that the increase in UC may be caused by some food additives. He stated, quote, possibly the cause of acute ulcerative colitis was connected with our food supply. Tinned or preserved foods may have something to do with it. Interestingly, saccharin was largely used in early years in canned foods to preserve vegetables, fruits, and meats, taking advantage of both its sweet and antiseptic properties. In 1901, Monsanto Chemical Company was founded for the sole purpose of producing saccharin. In 1903, Monsanto began shipping saccharin to Georgia to a little-known company called Coca-Cola. At the same time, strict regulation on saccharin importation, production, and use were put in many European countries like Germany, France, and Italy. The production of saccharin in Germany reduced from nearly 109 tons in 1901 to about 40 tons in 1903, among which only 3 tons were consumed within Germany. In 1907, a top food safety official of the United States tried to ban saccharin from use as a sweetener in the US, but the idea was ultimately opposed by President Theodore Roosevelt, who suffered diabetes and used saccharin regularly. During World War I, the shortage of sugar caused a great demand for saccharin. The figure here demonstrates the dramatic increase of saccharin consumption in Germany during World War I and II. During World War I, the ban on saccharin was lifted in Germany and other countries, accompanied by a striking increase in saccharin production and consumption. In accordance with the widespread use of saccharin since World War I, UC cases were also more frequently seen in countries other than the United Kingdom. As stated by Dr. Evans, he observed a large number of cases of colitis of citizens of different countries, including Turks, Arabs, and Indians during World War I, with many of them dying. In the United States, more cases of UC were seen since World War I. For instance, Logan reported 19 cases of UC treated in the Mayo Clinic before 1915, 18 cases in 1915, 23 cases in 1916, 57 cases during 1917 and April 1918, while Bargain reported that 693 chronic UC cases were examined in Mayo Clinic between 1923 to 1928. In another paper published in 1928, Dr. Solakson stated the greatly increased reports and attention of ulcerative colitis among the different countries during the past decade. Record high incidence of AD was seen during World War II. For instance, 525 cases of UC were diagnosed in the United States Army in 1944. with a rate as high as 12 per 100,000 in the 40 to 45 age group. This may relate to the preferred use of saccharin in the United States Army, which had chosen saccharin rather than sugar as the emergency ration for sweetening coffee or tea, taking the advantage of its immaterial weight as well as its antiseptic property. In the United States, Sigma Chemical Corporation was founded in 1945 to manufacture saccharin. In 1957, Svitlo was introduced. In 1958, Diet Red, the first diet soda, was introduced. In 1963, Tab was introduced into the market by the Coca-Cola Company. Both Diet Red and Tab were sweetened by saccharin and cyclamate. In 1969, cyclamate was banned due to its carcinogenicity in animals, and saccharin became the only artificial sweetener in the market in the US. In 1977, scientists in Canada found that saccharin caused blood cancer in rats. It caused fear all over the world about the safety of saccharin. The FDA proposed to ban the use of saccharin. However, this was strongly opposed by users of saccharin. At that time, saccharin was the only artificial sweetener. And last, Congress passed a 2-year moratorium preventing the immediate ban on saccharin, requiring additional research and a warning label. After multiple renewals of the 2-year moratorium, saccharin was eventually removed from the list of carcinogens in 2000. We may wonder how these saccharin-related events correlate with the incidence of IBD. Fortunately, there was a study by Stone and colleagues that collected data of new cases of IBD in Morrow County, New York from 1920s until 1986, as shown in this figure. We can see the dramatic increase of IBD after World War II correlated with the expanded market of saccharin and the introduction of the series of new products. While the mysterious decrease of IBD since the late 1970s correlated with the finding of the carcinogenicity of saccharin in 1977. Quantity data would be needed for more precise analysis. Unfortunately, I cannot find the information of saccharin use in Morrow County, but I indeed found some data on saccharin use in the United States. We can see a good match in the pattern of change, as well as a very high correlation between IBD and saccharin consumption. As we know, IBD emerged that dramatically increased along with industrialization and modernization. The co-occurrence of the finding of saccharin carcinogenicity and the decrease of IBD in the late 1970s would be a strong indication of the possible causative association. So I checked more into this issue. It turned out the mysterious leveling off or decrease in IBD in the late 1970s and 1980s seems true, as it was not only shown in Morrow County, New York, but also in other cities of the United States, as well as in many other countries such as Canada, Denmark, Sweden, Germany, UK, Japan, and Israel. Studies on the long-term change of IBD in Olmsted County, the US, or Copenhagen, Denmark, indicated the leveling off or decrease in IBD during this period was temporary, as it was followed by a later rebound. Here is the age distribution of the occurrence of IBD and saccharin consumption during the 1970s. We can also see the very similar pattern between these two factors. IBD is high in North America and Europe, but much lower in some other industrialized countries like Japan. Here we can see saccharin consumption in Japan was also much lower. This evidence made me believe that saccharin may be an important causal factor for IBD, so I wrote a manuscript in 2001. At that time, Nature just published 2 papers with funding by American and European scientists that the NOD2 gene, also known as CAD15 or IBD1, is highly associated with IBD. I felt the mystery of IBD may be soon resolved, so I submitted the manuscript to Nature, but was soon rejected. Then I submitted to more specialized journals such as Gastroenterology, that was also rejected. And last, the paper was eventually published in Medical Hypothesis. After I found the possible link between saccharin and IBD in 2001, I started to contact some IBD professionals as well as some national and international agencies such as the Crohn's and Colitis Foundation of America, NIH, FDA, EU, joint FAO, WHO expert committee on foreign additives in the center, advocating for them to conduct more research in this area. I got a response from Canada Health Agency. They told me that the use of saccharin had been greatly restricted in Canada since 1977, after the finding of a carcinogenicity of saccharin in animals. However, recent reports showed that Canada had the highest incidence of IBD in the world. These facts in Canada weakened my view on the importance of my hypothesis, as any hypothesis had to be tested against the facts. A decade later, in 2011, I found a paper showing that Splenda can alter gut microbiota. I immediately realized that it may share some similar effects as saccharin. Then I looked more into Splenda and learned that it is also a high-intensity, low-calorie artificial sweetener with an active ingredient known as sucralose that was first approved in Canada in 1991, followed by other countries. I wondered if the first approval of sucralose in Canada had contributed to the recent report of the record high incidence of IBD in that country. I looked into the temporal change of IBD in Canada and indeed found the possible link. For instance, IBD in Quebec had been lower than many of the other Western countries. As in the paper shown here, published in 1988, it was stated that, quote, in Sherbrooke, Quebec, both the incidence and the prevalence of Crohn's disease were low as compared with many other published studies. This was surprising, since high prevalence and incidence of figures are generally described for the Northern Hemisphere. However, studies in the early 2000s started to report world-record high IBD in Quebec. As stated in the abstract of the paper shown here by Marshall et al. Published in 2010, quote, with more than 20 cases per 100,000 person years, the province of Quebec, Canada, is among regions of the world with the highest reported occurrence of CD in relation to other places where comparable estimates are available. From the study by Low et al. Published in 2009, we can see a dramatic increase in the prevalence of Crohn's disease in Quebec starting since the early 1990s. From study by Beaton et al. Published in 2014, we can see autocolitis showed a similar pattern of change as Crohn's disease. I wrote a paper entitled, What Made Canada Become a Country with the Highest Incidence of Inflammatory Bowel Disease? Could sucralose be the culprit? And published in Canadian Journal of Gastroenterology. Interestingly, multiple reports describe a recent leveling off or decline of IBD in Quebec and other places in Canada. These changes closely correlate with the approval of sucralose in Canada in 1991 and the situation of the marketing in recent years, as discussed in my comments. The dramatic increase in IBD in the 1990s was not only shown in Quebec, but also other places in Canada, such as in Alberta. As stated earlier, after sucralose was first approved in Canada in 1901, it was approved in Australia in 1993, in the US in 1998, and in Europe in 2004. We can see remarkable increases in IBD after its approval. So I published a paper in the Journal of Inflammatory Bowel Disease suggesting sucralose may be an important causative factor for the puzzling recent worldwide increase of IBD, as shown in both developing and developed countries. As we know, at present, mainstream research still regards IBD as a disease with unknown cause, largely because none of the so many suspected or well-studied genetic and environmental factors can well explain the puzzling temporal and geographic changes of IBD. Back in 2001, people were very excited by the finding of the strong link between NOD2 and IBD, but it turned out that its correlation with population IBD is very poor. Studies can also suggest that antibiotics could be a risk factor for IBD, and that smoking increases the risk of Crohn's disease. It is reported that the use of antibiotics per capita in China is more than 5 times that of the developed countries. And China has a very high rate of smoking. However, IBD and CD in China remain very low. The poor correlation would just be the result of the minimal impact of not two antibiotics and smoking on IBD in the general population. In contrast, as shown in the slides above, marketing and the use of sedatives such as saccharin and sucralose correlate very well with many of the peculiar changes of IBD, such as its emergence in the late 19th century, its spread since World War I, its dramatic increase after World War II, its temporary leveling off or decrease during the late 1970s and 1980s, and its new round of worldwide increase in recent years. We all know that IBD may be affected by many factors, but these four actives seem to be among the predominant ones with huge impact on the population IBD, likely due to both their potent effect on gut bacteria and their wide use. The simple explanation of many puzzling changes in IBD would also suggest the cause and thus the mechanism of IBD could be simple. This new evidence led me to regain the confidence in the hypothesis I proposed a decade earlier, and bring forth a unified hypothesis on the etiology of IBD, including the cause and the mechanism of IBD, as well as the relationship between ulcerative colitis and Crohn's disease. In the paper, I discussed the many factors that may be associated with IBD, and provided my explanation on the etiology of IBD. Due to the time limit, I will not be able to elaborate in great detail. Here are the key points of the unified hypothesis. In general, I believe impaired innervation of a digestive process due to reduction in gut bacteria in modern society, along with improved hygiene and inhibition by food additives such as saccharin and sucralose, antibiotics, etc., and the resultant damage to the gut barrier and the tissue have played a primary role in both ulcerative colitis and Crohn's disease. The difference is that because of the large amounts of gut bacteria in the large intestine, UC is mainly caused by the increased infiltration of bacteria and their components, leading to the recruitment of neutrophils and the formation of a crippled abscess, which is a hallmark of UC. While CD is developed in relatively sterile parts of the gut or condition, and is mainly caused by the increased infiltration of food residues and antigens from the gut lumen, leading to the accumulation of macrophages and the formation of inflammatory granulomas, which is a hallmark of CD. Thus, UC and CD would be just shifting the manifestations of the same problem. Impaired innervation of a digestive process and damage to the gut barrier and tissue, along with the decrease in gut bacteria, were first manifested as UC, while further reduction in gut bacteria will make a shift toward CD, as manifested by the delayed appearance but a gradual catch-up of CD over UC in the population, the increase in chronic CD over time, and the more pronounced increase in CD by antibiotics and other potent inhibitory agents of gut bacteria. After the publication of that paper in 2012, more studies came out that provided more evidence of support. Here's a comment on paper published in 2012 showing increased pediatric IBD in Ireland since 2006, similar to the remarkable increase in pediatric IBD in Norway during 2005 and 2007, after the approval of sucralose in Europe in 2004, as discussed earlier. From this figure, we can see a continuous increase of UC, but a first up, then down of CD and IBD unclassified or IBD-U. Mainstream research may look at these changes as a big mystery and attribute it to the greater complexity of the disease, and may attempt to subdivide IBD into more subclasses as suggested in multiple publications in this area. However, these pattern changes can be easily explained by the unified hypothesis, which proposes that UC and CD are just shifted manifestations of the same problem associated with the progressive decreases of gut bacteria. Thus, even a single agent may cause shifted manifestations from predominant UC to predominant CD, depending on the extent of exposure and impact on gut bacteria. The decrease in CD and the increase in UC shown here may be just caused by a decreased exposure to the causative agent, along with the shifted market. Food additives such as sucralose are used in thousands of beverages foods, etc. The complicated and dynamically changing patterns of formulation, marketing, and consumption of food additives may be the simple main cause of the puzzling irregular temporal and geographic changes of UC, CD, and IBD, as shown here and in many other studies. Here is a paper published in 2013 showing the nationwide increase of IBD hospitalization in the US from 1997 to 2009. From the study on IBD in North California from 1996 to 2006, we can see the increase shortly after the approval of sucralose in 1998. So I published a paper with a discussion on when and how was the new round of increase in inflammatory bowel disease in the United States studied. In 2017, a paper was published and provided a more complete picture of the temporal change of IBD in Olmsted County, Minnesota from 1940 to 2010. We can clearly see a recent rebound of IBD after a temporary decrease in the 1980s. At present, many people attribute the remarkable increase of IBD in developing countries to the adoption of the westernized lifestyle that may involve numerous factors. The ups and downs of IBD, even in the well-developed Western countries such as the US and Denmark, as shown here and in previous slides, would suggest the existence of some main environmental factors that are closely associated with IBD. These mysterious ups and downs can be easily explained by dynamic changes in marketing and use of food additives such as saccharin and sucralose, suggesting the reason of these ups and downs, thus the cause of IBD, could be much simpler than people perceived. Here is a comment on a study published in 2013 showing the remarkable increase of pediatric IBD in Singapore after 2000. Singapore is a well-developed, modernized country. A sudden increase in IBD would suggest IBD is not simply associated with modernization, but rather some simple events associated with modern life, such as the new approval of the artificial sweetener sucralose. Here is a large-scale epidemiological study published in Gastroenterology in 2013 on incidence of IBD in the Asia-Pacific region. Guangzhou, a city in South China and not far from Hong Kong and Macau, showed the incidence of IBD as high as 3.14 per 100,000 persons, which is much higher than that of the northern cities in China, such as Chengdu and Xi'an, both being just at around 0.5, and also much higher than the more developed Hong Kong, Macau, and Singapore, which had an IBD incidence at 2.62, 2.24, and 0.97 respectively. This result suggests the existence of some environmental factor that is strong enough to negate and overturn the general trend of a higher risk of IBD in northern and southern locations and in developed or developing regions. Guangzhou is the capital of Guangdong province, while sweetness is the primary feature of the Guangdong cuisine. I wrote a comment with my simple explanations that the wider approval and use of food additives such as sucralose may have contributed to the narrow gap of IBD between the developed and the developing countries, while the puzzling high IBD incidence in Guangzhou may be simply associated with their preferred taste in sweetness. This notion is supported by the multicenter study on risk factors of IBD in China, published in 2013, with the finding by the multivariate analysis that heavy sugar consumption was a key risk factor, while no significant association were found between IBD and factors such as smoking, appendectomy, breastfeeding, a history of measles, rural or urban residents, education, oral contraceptives, and the use of non-steroidal anti-inflammatory drugs or NSAIDs. Here we can see the exceptional close relationship between IBD and sweetness that surpass many of the well-studied risk factors of IBD. I wrote a comment suggesting that IBD may be associated with artificial sweeteners rather than sugar, as evidenced and discussed earlier. Here is a comment on study showing the emergence and the striking increase of IBD, especially Crohn's disease in Saudi Arabia since early 2000. Again, this abrupt increase would be likely caused by the introduction of some agents, such as a new approval of sucrose, while the predominant increase in CD over UC would be just the result of high intake of the causative agent by the exposed population and high impact on gut microbiota, similar as the pronounced increase of CD with antibiotic usage, as discussed in the unified hypothesis above. IBD may be prevented if we found out and eliminate the cause. Here is a comment on studies showing the dramatic increase around 2000, but a recent leveling off of IBD in South Korea. The recent leveling off of IBD in this industrialized country suggests again IBD may be stopped once we found out and eliminate the cause. Here are some of my other publications commenting on the findings across multiple studies to address issues such as food additives may be the possible cause for recent remarkable increase of IBD in children, how to explain the discordant change of ulcerative colitis and Crohn's disease in adjacent or even the same regions and time periods, and why pediatric IBD in Sweden shared a similar trend of change as a general population IBD in Denmark, but not pediatric IBD in Norway, as explained by the different pattern of use of food additives, such as various artificial sweeteners. In 2014, Nature published a study showing artificial sweeteners induce glucose intolerance by altering the gut microbiota. The high impact of the journal finally brought a lot of attention to the possible adverse effect of the artificial sweeteners on gut microbiota and health. It showed the adverse effect of artificial sweeteners can be alleviated by antibiotics to both gram-positive or negative bacteria, suggesting that the defects of some general function of the microbiota, rather than changes in some specific species of gut bacteria, may be the cause. Another paper published in 2015 in Nature shows that low concentrations of two commonly used emulsifiers induce low-grade inflammation and obesity metabolic syndrome and promoted robust colitis in mice as a result of changes in gut microbiota rather than a direct effect on gut mucus and gut barrier. Putting these two papers together would indicate that a general change of gut microbiota by either artificial sweeteners, emulsifiers, or something else may potentially increase the risk of colitis and other health problems, and suggested in the series of my publications, such as the two listed here, published in 2002 and 2007. In my papers, I provide a simple mechanistic explanation as to how artificial sweeteners, microbiota, and IBD are associated with each other. Unfortunately, the Nature papers presented the relationship between gut microbiota and health as extremely complex and a big mystery, with complete ignorance of the simple explanation that had already existed for many years. This reflected the general status of microbiome research. In recent years, microbiome has become an extremely hot area of research. We have gotten enormous information about the composition, structure, functional pattern, environmental distribution, differences among different species of animals and different stages of evolution, and human microbiome at different locations among those with different ethnic background, economic and living conditions, geographic locations, diets, disease conditions, etc. People are overwhelmed by the complexity of the microbiome. At present, the relationship between gut microbiota IBD and other diseases is presented by mainstream research as extremely complex and desirable only by sophisticated approaches such as the multiple omics like genomics, transcriptomics, proteomics, metabolomics, epigenomics, etc., and metaomics, to which I have totally different view. As I discussed earlier, I believe the primary cause and the root mechanism could be simple. Here is a paper I published in 2015 to impress my view. The citations highlighted in red are the series of publications in Nature and related journals on risk genes of IBD. Since finding the first risk gene, NOD2, in 2001, till recent findings that about 200 genes are significantly associated with IBD. As we know, IBD emerged and dramatically increased for just about a century, suggesting some environmental but not genetic factors have played the predominant role. Despite that, the finding of the first risk gene of IBD in 2001 was followed by ardent pursuit by elite professionals in this area, with a series of publications in the most prestigious journals. Not surprisingly, it turned out that none of these genes alone or in combination can really explain the mysterious change of IBD. However, the finding of these risk genes were presented as a series of great achievements, and their failure to truly explain the cause of disease was attributed to the greater complexity of the disease itself, making any simple explanation unhealthy. The citations highlight in blue are the series of my explanations of the different aspects of IBD, also since 2001. Despite my persistent advocating since 2001, all these evidences and explanations remain largely ignored by the mainstream research. I feel a great tendency of fetch chasing rather than problem solving in current medical research. We should realize that many of the changes revealed by omics and metaomics would be just the consequence rather than the cause of the disease, while the crucial event may be subtle, transit, and simple. The superficial explanation of data could be misleading or even detrimental for the solution of the disease. In my opinion, we should track down the root toward the primary cause of the disease, rather than just explore up and likely got lost among the small branches and the leaves. In 1991, when I was a postgraduate student, I wrote a paper entitled The Philosophy in Life Science, the Phenomenal Complexity and Diversity and the Essential Simplicity and Unity, and published in Medicine and Philosophy. I had philosophy then, and I hold the same philosophy now. There are millions of species of life on Earth, but all the differences arise from the different arrangement of just several nucleotides on the polynucleotide chain. In the era of big data, machine learning, and advanced technology, great efforts can be made to explore the complexity of the microbiome and its relationship to health. But in my opinion, we should see through the phenomenal complexity and find the potential shared simple cause and mechanism. Here is my explanation as to how impaired inhibition of the digestive process may play a critical role in IBD and other health problems associated with dysbiosis of gut microbiota. I believe a decrease of gut bacteria in modern society, along with improved hygiene and inhibition by food additives and biotics, etc., cause a decreased formation of decaying bilirubin and impaired inhibition of digestive process in lower gut, which cause damage of gut barrier gut tissue, resulting in the leaky gut and IBD, while the increased infiltration of bacterial and other toxins from the gut lumen into the blood will lead to the development or worsening of many diseases of the whole body. You may find dozens of my publications by searching my full name XIAOFA space QIN on PubMed. Unfortunately, only publication after 2004 can show up, so my first publication in this area, written in 2001, cannot be found that way. Nevertheless, you can find a series of publications with my view on many issues of IBD and gut microbiota, such as the pathogenesis of IBD, the NOT2 gene, autophagy, and the genome-wide association studies, the relationship between ulcerative and Crohn's disease, the relationship between primary sclerosing cholangitis and IBD, the relationship between IBD and irritable bowel syndrome or IBS, the association of IBD with many other factors such as hydration condition, antibiotics, probiotics, dietary chemicals, enteral and parenteral nutrition, meat, Yoni disease, and Mycobacterium avium subspecies perituberculosis syndrome. You may also find a series of collaborative studies on conjubilubin, bilirubin, saccharin, and sucrose in animal models of colitis and bile duct ligation. Here are some recent papers that you may be interested in. One is a discussion that dysbiosis caused by dietary chemicals such as sucralose and saccharin may be more detrimental to the gut and health than antibiotics. At present, many people may think antibiotics have the most potent adverse effect on gut microbiota and health. With the evidence I collected and my analysis, I suggest that dietary chemicals such as saccharin and sucralose could be more detrimental. Antibiotics are mainly only briefly used by patients, while sedatives are used daily by many people of the general population. In addition, the potent inhibition on gut bacteria by antibiotics may also reduce the amount of endotoxin, as well as the glycosilysis that degrades the protective mucus. Thus, high inhibition on gut microbiota may not parallel with high adverse effects. As shown by the study published in Nature on artificial sweeteners that further inhibition of gut bacteria by antibiotics to either gram-positive or negative bacteria alleviated rather than exacerbated the adverse effect of the artificial sweeteners like saccharin. This notion is also in accordance with the phenomenon pronounced promotion of growth of farm animals such as pigs, cattle, etc., by fitting different antibiotics and a sub-therapeutic dose that may be just 1/50th of the therapeutic dose. This daily use of such low doses of antibiotics may share a similar impact on gut microbiota as the consumption of sedatives such as saccharin and sucralose in the general population, rather than the treatment of patients with antibiotics. As discussed earlier, shortly after its marketing, saccharin had been used as antiseptic agents to treat some infectious problems such as thrush and diarrhea. Artificial sweeteners such as sucralose, cyclamate, and ACE-CFMK showed a similar impact on bacteria as saccharin. Not surprisingly, these artificial sweeteners have also been used by many farmers in livestock and achieved the same effects of increases in growth rate and feeding efficiency as sub-therapeutic dose of antibiotics. Another one is a discussion that non-nutritive sweeteners may have a bell-shaped dose response for dysbiosis-related adverse effects. Their peak adverse effect may be well below the current acceptable daily intake or ADI. A combination of the high effect and the low intake and the wide use would potentially make these sedatives capable of exerting big impact on the general population. The third one is a discussion on paper recently published in Gut by Canadian scientists showing increased fecal proteases before the onset of ulcerative colitis. They thought the increased proteases are from gut bacteria. I showed evidence that the increased protease activity would be more likely the result of impaired innervation of proteases from the pancreas. In fact, I had published a paper in 2014 to address this issue. I had been engaged in medical research in universities in China and the US for more than 20 years. Several years ago, I left academia and started a company aiming at developing products to solve some problems such as IBD and other health issues associated with dysbiosis of gut microbiota. After years of effort, GI Biopharmic developed the first product, GI-Z01 Probiotics, as a probiotic dietary supplement, which can be purchased online at the following websites. It showed more efficacy than fecal microbiota transplantation or FMT in an animal colitis model. It warrants further study. More research and development are underway. Here is my contact information. Welcome to collaborate. Thank you very much for your attention.